Good morning, good afternoon, and good evening. My name is Delvin Wenani, and I'm a biology teacher. And today I want to take you through the Kcse essay questions from Form 1 syllabus. And therefore, remember to subscribe my channel. Remember to follow, remember to comment, share, so that we can reach as many people as possible. I'm doing this in 4 series, so I'll be doing the part 1, and in part 1 I'm talking about the Form 1 essays. I'm talking about the Form 1 essays, and these are some of the questions that come from Form 1, which are asked as essay question. We understand paper 2, question number 7 and 8 is an essay kind of a question, and therefore you are requested to write in an essay form. So ladies and gentlemen, without wasting much time, let us start. Now, these are some of my suggestion questions that could have come as an essay question in Form 1. Number 1, explain the various ways in which a typical cell is adapted to its function. So we are talking about the adaptation of the cell to its function. So a cell is adapted in the following way. So we shall be looking at some of the features that it has and how they are adapted to the function that they perform in that cell. One is the presence of a cell membrane. A cell has a cell membrane, and this cell membrane has pores, and therefore we call it semi-permeable. That means it allows or regulates the substance entering and leaving the cell. Number 2, it has a cytoplasm. Now, the cytoplasm, this one now contains the sugars and the salts. And also the cytoplasm maintains what we call the osmotic pressure and also act as a liquid medium for all the biochemical reaction that takes place within that cell. Number 3, the cell has a nucleus. And the nucleus contains, it contains chromosomes, which in nature they have the hereditary materials and controls all the activities of the cell. Number 4, the cell has ribosomes, and ribosomes are site for protein synthesis. It also has the Golgi bodies, or what sometimes the books call them the Golgi apparatus, which are for secretion of hormones and enzymes, and also formation of lysosomes. It also has lysosomes, and lysosomes contain lytic enzyme for breaking down what we call worn-out cell organelles. Worn-out cell organelles. The word worn out, in simple terms, we talk about old, aged, or we can talk about the aged and old, the tired ones. So they are destroyed and broken down. Not necessarily being destroyed, but they are broken down. Breaking down means they are broken into another substance, maybe for easier transportation, for elimination. It also has secretory vesicles, which are formed from Golgi apparatus for secreting substances. It has a smooth endoplasmic reticulum, which synthesizes and transports lipids. The cell also has what we call a rough endoplasmic reticulum, and this one they transport proteins. It also has a nucleolus. Now, nucleolus controls the activities of the nucleus. So we had talked about the nucleus having the hereditary materials and also control the activity of the cell, but nucleolus controls the activity of the nucleus and also produces what? Ribosomes. It has a mitochondria, and this forms site for energy production, or what we call it forms sites for respiration. And we know the end product of respiration is energy. And therefore, talk about form site for energy production. It has centrioles. Centrioles, they help in the formation of cilia and flagella. Cilia and flagella. and also form spindle fibers, which are used in cell division. When we shall reach Form 4, we shall be talking about the cell division. And therefore, that's when I'll bring in the issue of spindle fibers, which are formed by the centrioles present in a cell. It also has a plant subvacuole, which stores salts and other dissolved substances, and also control the osmotic pressure and turgidity of the cells. Food vacuoles, they are present, and the food vacuoles, they are involved in digestion of engulfed food. It also has a chloroplast. These plant cells have chloroplast, and the function of the chloroplast is they form sites for photosynthesis in a plant cell. Question number 2 that may come as an essay is, explain how the various specialized cells are modified to carry out their functions in plants and animals. And these are, we have animals. Animals, we have cells like the sperm cell, which has acrosome. The acrosome is found on the head region, and this acrosome has lytic enzymes that digest the egg membrane for easy penetration during fertilization. Number 2, it has a long tail. That long tail contains numerous mitochondria to generate maximum energy for propulsion and also swimming in the what we call the vaginal fluid after ejaculation. The red blood cells. Red blood cells, these are flattened, circular, sometimes spherical, by concave in shape, and their function is to increase the surface area for packaging of hemoglobin. It also has hemoglobin that combines with the respiratory gases for transportation to and from the body tissues. It has also white blood cells, and the function of the white blood cells, these ones, they are amoeboid in shape. When we say amoeboid in shape, we mean they are able to change shape, and therefore they engulf pathogens through a process known as phagocytosis. They phagocytose through a process known as phagocytosis. It also has lymphocytes. In the white blood cells, we have lymphocytes which produce antibodies to fight pathogens. We have nerve cells, and the nerve cell, these are extensions, or they have extension, or we call them the dendrites, to receive and send information for sensation. We call the impulses. And they are also ciliated, and they have what we call ciliated epithelial cells. They have ciliated epithelial cells. They also have cilia for propulsion of mucus that traps dust and microorganisms in the respiratory tract. Muscle cells, they are elongated, some are striated and contractile. And this is all about to bring movement. Remember, muscles bring movement in terms of contracting and relaxation. In plants, we have the following cells. Number 1 is guard cells. And these one, they are bean-shaped. Talking about, they look like bean. And you can see from the photo, they look like beans. And this is to regulate the size of the stomata, and also allowing gaseous exchange and control water loss. The opening and closing of the stomata affect the transportation of water in terms of water losses, that is through what we call transpiration. It also has chloroplast. Now, chloroplasts have chlorophyll, and this basically is for photosynthesis. Root hairs, or what we call the root hair cells. And this one, they are elongated, thin-walled. We talk about being elongated. First of all, they are elongated, thin-walled with dense cytoplasm. And this is what? Absorption of water and mineral salts. We have epidermal cells. Epidermal cells, they are thin for protection of inner tissues from mechanical and microorganism attack. Next one is palisade cell. The palisade cell contains numerous chloroplasts with chlorophyll. And this one is basically for photosynthesis. And they are also elongated to increase surface area for trapping maximum amount of light energy. They have meristematic cells, and the meristematic cells here, they are thin-walled with dense cytoplasm, and their function is for primary and secondary growth. We shall talk about that one when we reach Form 4, and we are looking at the essays in Form 4, and we shall talk about growth in, that's the primary and secondary growth in plants, and we shall look at how the meristematic cells bring about this primary and growth in those plants. Number 3, how are the leaves of higher plants adapted to their functions? 1, they are broad and flattened lamina. They have broad and flattened lamina. That is the part of the leaf. Now, this is to increase surface area for absorption of light. To increase surface area for absorption of light. They also have a thin blade, and this is to reduce distance for diffusion of gases and penetration of light waves. They are also transparent epidermis. They have transparent epidermis and cuticles. We talk about some plants like, for example, the one which are found in arid and semi-arid areas, that they have issues to do with the cuticle. Why? Because cuticle plays a key role in allowing light. And therefore, here we are saying it has a transparent epidermis and cuticle to allow light to penetrate to tissues. The cuticle layer is absent on stomata to allow for gaseous exchange. They also have one cell thick epidermal layer, and this is to reduce the distance over which sunlight penetrates. To reduce the distance over which sunlight penetrates. Palisade cells have numerous chloroplasts which containing chlorophyll to trap maximum amount of light energy. They have stomata on the epidermis. Why? To allow for gaseous exchange and control of water loss through transpiration. Palisade layer, they have a palisade layer which have elongated cells located at the right angles to the leaf surface. Why? For maximum absorption of light energy. They also have a spongy mesophyll which consists of spherical and loosely packed cells. Now, this is to create air spaces which communicate with the atmosphere through the stomata. And this is for the purpose of gaseous exchange and control of water loss. It has veins, and these veins have conducting tissues. Those are the xylem for movement of water and dissolved mineral salts. And also it has phloem, and the phloem is for translocation of manufactured food from the site where they are manufactured to the rest of the plant. Number 4, explain how the various teeth adapt mammals for nutrition. Number 1, we have the incisors. Incisors, they are sharp, chisel-shaped. Sharp, chisel-shaped, and this is for biting, cutting food, and remember, they also have one root for support in the jawbone compared with the rest. We have the canines. Now the canines, they are long. These are long, they are sharp and pointed. And majorly, they are for holding prey, piercing it, and tearing flesh from prey. They are single root, and this one is the single root offers them support in the jaw bone. We have the premolars. The premolars, they are large, wide. Talk about the largeness is to increase surface area for grinding food. They are cusped to increase surface area for grinding food. They have two roots for firm support and anchorage in the jaw bone. Molars, they are wide, large, to increase surface area for grinding food. Remember, between the molars and premolars, they are very close, but the molars are highly cusped. They are highly cusped to increase surface area for grinding food. Number 5, describe what happens to a meal of oily beans and maize from the time of ingestion up to the time of absorption. We start in the mouth. The digestion starts in the mouth. 1, there is starch in the maize because remember it's a mixture of maize and beans. It's an oily maize and beans meal. So digestion begins in the mouth, and in the mouth, starch in the maize is digested by the salivary amylase. We call it amylase, you can call it tyalin, you can call it diastase, into maltose. Food is also chewed and mixed by teeth and tongue. It's rolled, the process of chewing and rolling, and it forms what we call a bolus. Now, this bolus shall be swallowed through a process known as peristalsis, and it will enter into the stomach via the cardiac sphincter. In the stomach, the entry of the bolus in the stomach shall trigger the production of gastric juice. Now, the gastric juice contains pepsinogen. Pepsinogen shall be activated into pepsin. Pepsin is the active enzyme from pepsino. Pepsinogen is an inactive enzyme. And this enzyme is meant to digest proteins in the beans into shorter, what we call the peptides. Then the food shall be churned and allowed into the duodenum via the pyloric sphincter muscles. Once the food reaches the duodenum, the bile juice shall be secreted by the gallbladder, and this emulsifies the oils in the beans into tiny droplets through a process known as emulsification. There is also production of pancreatic juice, which shall be secreted by the pancreas. The pancreatic juice contains pancreatic amylase that will now proceed with the digestion of starch, which had stopped once the food entered the stomach because of the presence of HCl. And this now will continue digesting starch to maltose. The pancreatic lipase now that has been produced together with the pancreatic juice shall now digest the oil in the beans to fatty acids and glycerol. We also have trypsin. Now, trypsin shall digest proteins into shorter peptides, the process that was taking place in the stomach. Then, from there, the food shall enter into the ileum, and where now we have succus entericus is secreted, and this one contains maltose enzyme. Now, the maltose enzyme, we call it the maltase, and maltase shall digest maltose into glucose for easier absorption. We also have peptidase, and peptidase shall digest peptides into amino acids. We also have lipase, which digest the remaining lipids or oils into fatty acids and glycerol, which is then absorbed through the lacteoles of the villi. How are the small intestine in mammals adapted to their functions? How are the small intestine in mammals adapted to their functions? Number one, small intestines consist of the duodenum and the ileum. They are together. And this is where most of the digestion occurs. Bile from the gallbladder, that is from the liver, is secreted through the bile ducts, and it is used to emulsify fats. I talked about breaking down fats into smaller, tiny droplets. And why are we breaking the fats into smaller droplets? It's for easier enzyme activity, so that enzymes can act on the food easily. In other terms, we say it is to increase the surface area for enzyme action. We also have the pancreas, which secretes pancreatic juice. Now, the pancreatic juice is also secreted in the duodenum. And the pancreatic juice contains, number one, it has pancreatic amylase. The pancreatic amylase helps in breaking down the remaining starch into maltose. Remember, digestion started in the mouth. Once the food was digested by the salivary amylase, during swallowing, once the food enters the stomach, there is production of gastric juice, which has HCl in it. And therefore, this one stops the further digestion of starch because of the alter in the pH. That's the lowering of the pH. So now when the food enters the duodenum, which is now in the part of the small intestine, what happens again? There's production of sodium hydrogen carbonate. And this one will neutralize the acid. So it gives room for another amylase to work on starch that's remaining. And that's where pancreatic amylase comes in to break down the remaining starch. So another digestion of starch is done in the duodenum by the pancreatic amylase, which breaks down the remaining starch into maltose. We also have trypsin that is secreted in active form. We talked about trypsinogen, and activated by enterokinase enzyme, which hydrolyzes protein into shorter peptides. We also have pancreatic lipase, which converts lipids into fatty acids and glycerol. We also have sodium hydrogen carbonate. It's also produced in the duodenum. And the major reason why it's being produced is to neutralize the acid chyme from the stomach and provide a suitable alkaline medium for pancreatic and other intestinal enzymes. Also, we need to understand that the ileum is long and narrow. And why is it long and narrow? This is to increase surface area for complete digestion of food and the maximum absorption of digested food. It is also very highly, you can use the word highly coiled. And this is to reduce speed of flow of food. And this is for maximum digestion and absorption. It also has villi and microvilli, and this is to increase surface area for maximum absorption. It has dense network of capillaries. Now, this is for efficient transportation of absorbed food. Presence of the lacteose in the villi, and this is for absorption of fatty acids and glycerol molecules. Presence of enzymes, I've mentioned them. For example, the lipase for digestion of lipids into fatty acids and glycerol, maltose for digestion of maltose to glucose molecules, Peptidase for breakdown of peptides into amino acid, sucrase for digestion of sucrose into glucose and fructose, lactase for digestion of lactose into glucose and galactose. It also has what we call goblet cells. And these goblet cells, they produce mucus. And the function of the mucus is to lubricate the walls of the ileum for smooth flow of food, and also coat the wall of the ileum to prevent digestion by peptidase enzyme. So ladies and gentlemen, that one brings us to the end of part 1. I'll be back with part 2. Just remember to subscribe, let us remember to share so that you can reach as many people as possible. Otherwise, be blessed and stay safe. God bless you, and until we meet again, asante sana.